We are presenting on taxonomic classification of who-defined viral pathogens in complex metagenomic datasets. For the classification, we will be using various machine learning architectures as multi-class classifiers. This is our outline and the format of our presentation. In this presentation, we want to highlight how the world's current outbreak detection is slow, but can potentially be improved with machine learning. In order to demonstrate that current outbreak detection is slow, the figure on the right shows the steps of the current process. The entire process takes, on average, 20 days from onset to outbreak classification. We are hoping that our work will quicken this process. For an example, COVID-19 began when the first symptoms were detected in December 31st, 2019. Officials declared the COVID-19 outbreak a global health emergency on January 30th, 2020, and a global pandemic on March 11th. But the most important date was January 12th, when the first SARS-CoV-2 genome was published. If a technology only needed the genome or parts of a genome to detect when large populations had been affected, the pandemic could have been far better handled, potentially mitigating the damage it caused. In our presentation, we hope to demonstrate the ability that machine learning has to solve this problem. Furthermore, we want to detect more than just one virus. We want to detect multiple species. In our work, we focus on the 8 who-defined viral pathogens of interest. To detect all the species we are interested in, we assume that we have access to wastewater samples from a local wastewater treatment facility and that we sequence the genomes from organisms within the samples. In this way, we can detect how infected a community's population is. In order to train our models, we feed our models with 2 basic data sets, GenBank and RefSeq. RefSeq is considered the higher quality data set than GenBank, as it is non-redundant and curated. This assumption is something we examined in our work, and we also hope to bring insight on. In order to test the quality of the databases, we alternate the data sets into our models as either the training or testing data set to see how one performs on the other. Our pipeline was as follows. It begins with the previously discussed databases, downloading their genomes, splitting them into the train and test data sets to ensure unique genomes for each data set, creating perfect 250 base pair unique fragments, encoding the fragments into a machine-readable format, and finally training the models. We will first focus on the encodings. An important factor to consider when we try to feed the neural network with biological data sets is the encoding type. A common way of doing this is the one-hot encoding on the left, where we represent each nucleotide as its own class. However, we introduce a new encoding, the codon action tensor coding, also known as cat encoding. Instead of representing it by individual nucleotides, we represent them as a group of 3, where the codon is the simplest unit of DNA to be interpreted by organisms. We implement our networks based on 3 different models. First is the basic CNN only. Second, the LSTM only. And third, the CNN plus LSTM hybrid, the most common architecture of choice in the literature. We want to focus on the main findings of the research, and the following are those findings. 1, GenBank is more representative than RefSeq in taxonomic classification. And 2, each model architecture has a corresponding optimal encoding. So first we compare over our 2 data sets. As you can see from the graphs, the GenBank best result is represented by the solid line, while the RefSeq best result is represented with a dotted line. From the graphs, you can see that the GenBank trained models perform better than their RefSeq counterparts, which contradicts the popular assumption that RefSeq is better to train on. Second, we compare our 2 encoding types, the one-hot encoding and the cat encoding. While we don't see a significant advantage over all models, we do find that LSTM models prefer one-hot encoding, as you can see here, and CNN-only models prefer cat encoding, as you can see over here. For the CNN plus LSTM model, it does not show significant preference over encoding types. This suggests that novel cat encoding can be used as a substitute for more popular encodings, as you saw with the CNN models. To emphasize the findings, we found that GenBank is more representative than RefSeq in taxonomic classification, and each model architecture has a corresponding optimal encoding. In the future, we can try to include Illumina, PacBio, or Oxford Nanopore sequencing simulated dataset as a potential dataset for training. In the future, we can try to include an Illumina, PacBio, or Oxford Nanopore sequencing simulated dataset as a potential dataset for training. For the dataset, we could vary our genome's fragment length and test different models afterward. Last but not least, we could try to tune and compare the model's hyperparameters of window size, some form of dropout, and the learning rate.